Good morning, children. Today we are going to discuss the topic classification. Its two aspects, phenetic and phylogenetic. So, there are two broader class systems, phenetic systems and phylogenetic system. So, the physical attributes and such phenotypic similarity or resemblances, when you are consider, such a system is known as phenetic system. So, that is also known as numerical taxonomy. Here, plants are classified based on their characters. Okay, some of the characters that we can see from outside. Based on that, we can classify them. Only some characters we are considering here. Clear? But what do you mean by the second system, that is cladistic or phylogenetic? Here, evolutionary relationship is considering. Okay, so mainly based on evolutionary relationship. So, pattern of phylogeny. Here we have to imagine an ancestor and from which how the modern system evolved. So, the phylogenetic relationship is considered in the second category, that is cladistic. We can discuss the both in detail. Okay. First one is numerical taxonomy. Numerical taxonomy or phenetic classification. So, numerical taxonomy, what it is actually? Plants are classified mainly based on their characters. It is clear. Okay. So, taxonomists usually classify plants based on one or two character only. One character usually. One example is dicotyledonnae and monocotyledonnae. How they are classified? Based on the number of cotyledons. Okay. So, here actually classification with emphasis on one character. That is known as a priori. Clear? Okay. But Michael Adanson, a famous taxonomist, Michael Adanson, first time pointed out equal weightage should be given for characters while classifying plants. So, you want to neglect characters. You have to accept, you have to give weightage to each and every characters. So, this branch is actually that latest in modern time, we can utilize the computers for doing this. This is also known as mathematical taxonomy, taximetrics, multivariate morphometrics, phenetics, etc. known by different names. So, it is actually the balance between an empirical approach and an experimental approach. You know what is both? Empirical and experimental approach. So, in those times only some empirical approach is there. Observation that we are made in conclusion only by some observations only. But now what we are doing? Some experiments also. So, besides this, these two are compiled in the system. So, it is a new method of organizing data. So, a combination of empirical and operational, or the second half operational or experimental approach. Okay. Now, what are the principles of numerical taxonomy? I already told this is proposed by Michael Adanson, and here equal weightage is given to all characters while classifying a plant. Okay. Here, actually what they did is, they rejected the idea of giving more importance to certain characters. So, usually our system is like that. So, one or two, gamopetalae means petals united, polypetalae, petals free. So, we are stress given to the union of petals only. But in the system, at the same time, we are also giving weightage to all other characters. Understood? Okay. So, all characters are taken into consideration. And this is again modified and modern numerical taxonomy that is developed by Sneath and Sokal in 1973. So, this system of modern numerical taxonomy is developed from the previous Adanson's system. So, the modern principle of numerical taxonomy is known as Neo-Adansonian principles. So, this Adansonian principles is very important. Usually come for examinations. Clear? What are the principles? First one is, greater the content of information in the taxa of a classification, the more the characters it is based, means the classification become good. Okay. So when you are having more information, when your content is very high, means you are considering large number of characters. Clear? So your classification will be good. Okay. Then a priori. Every character having equal weightage. Another important point is overall similarity between any two entities is a function of their individual similarity. Means overall similarity between any two entities. For example, phenotaxy, opposite world conditions, two conditions, and you can compare it in both cases in all its aspects, the phenotaxy. So and that constitutes the individual similarity. The distinct taxa can be recognized because correlation of character differ in the group of organisms under study. So correlations of characters differ. So when you take two groups, it may be of different character. Those who are related is different. Clear? Now, phylogenetic inferences is also made from taxonomic group, and taxonomy viewed and practiced as an empirical science, and classification is based on phenetic similarity. These are the neo-Adansonian principles. Okay, now what are the methodology of numerical taxonomy? How they are doing numerical taxonomy? Okay, so first of all we have to select all individuals and thereafter their characters are spotted out. Okay, then on the basis of character analysis, resemblance among the individual is established. So you have to collect all individuals, you select individuals, then study them on the basis of character analysis, resemblance, etc. noticed, and work out later stages with the help of computers. Clear? This is the main point, main steps. So selection of, there are some scientific terms for this. First I told selection of individuals. Here it is known as selection of operational units. Operational unit. Population, species or genera, or from which, anywhere you collecting information. That is the selection of operational unit. Then this information recorded and similarity is determined by statistical analysis. Then compare the similarity of data and construct a diagram and models. This is the three basic steps in numerical taxonomy. Okay. So what are the advantages? Here, power to integrate data from variety of sources. So you can depend morphology, you can depend anatomy, molecular biology, phytochemistry, cytology, from varieties of sources you can select. Then easy handling, even less skilled. So anybody can handle this very easily. Then another peculiar feature is data coded in numerical form is usually can be integrated and with existing data processing system and is used for creation of key, catalog, map, etc. Okay, it is an improved the conventional taxonomy we are now using. Okay, so it is an advanced one. Then provide a better classification and key. Number of biological and evolutionary concepts are reinterpreted through this system. Then it aims at determining phenetic relationship between organisms, and it is also arrangement by overall similarity and observation. So we are observing, then similarity measuring, weightage of characters. That is important. Okay. So phenetic relationship is similarity, resemblance based on phenotypic characters. So what we are using here is usually phenotypic character, number of characters under consideration, then we are comparing them or resembling them. Okay. Phenetic relationship is, finally it is represented by phenogram. Okay. But when you are considering evolutionary relationship, you can record it in the form of what? Cladogram. That is cladistic relationship. Here it is phenetic relationship. That is measured through observed by phenogram. Okay. So first I told the first step is what? Selection of taxonomic unit. That is operational unit. Here it is known as operational taxonomic unit. That may be from genera or family or species, anything. From the population you can select the individual, and that is your study. So first step, and it is sample from which data collected. That is known as what? Operational taxonomic unit, OTU. Okay. Now, second step is what? Character study. Maximum number of character that distinguishes one taxon from other. For example, you can have white flowers distinguish one species from another with the red flowers. Likewise, then that is a character that is vary from one species to one organism to another. So here flower color is a character, white and red are its two states. So a character and its states you can consider. Okay. So color is a character, flower color is a character, white and red is its states. Okay. Then it is important to select unit character for numerical analysis. Unit character. You have to stick on such a characters. That is very important. Now, what is an unit character? A character with two or more states. Example, trichome. What is trichome? Hairs. They are glandular or aglandular. Means those with the glands or without a glands. Then, in the glandular itself again there is what? Sessile or stalk. So, if all glandular trichome is the our operational unit or is same type, trichome is selected as unit character. Okay. If they are different, then glandular is unit character. See. So, if all glandular trichome is our same type, means what? All glandular trichome is same type, there is no branched or there is no multicellular, likewise all same, the trichome is a unit character. Clear? If they are different, then glandular is unit character. Means what? Under glandular different states are there. In trichome, glandular, aglandular. If glandular is same type, trichome is the unit character. If glandular is of different type, glandular is the unit character. Likewise it goes. Then all characters are weighted equally. Okay. Not less than 60 characters but more than 80 are desirable. So you have to select more than 80 but it should not be less than 60. Okay. For some characters, if information not available or irrelevant or show variation are omitted from the list. Such characters are called residual. Some characters, sometimes you cannot have clear-cut data or under observation you are not getting it. Such characters you can remove. You can neglect it. That is known as what? Residual weighting. Only homologous characters are being compared. Clear? Now, next step is what? Coding. First you selected operational unit, taxonomic unit. Second one you studied the character and its states. Third one is what? Coding of character. So here we need the modern technology, computers. Two states characters are usually good. You can have presence or absence. Two states, for example, glandular or aglandular, woody or herb, likewise. Then there are qualitative as well as quantitative one. Qualitative or multi-state are there. Flower with the white, yellow, orange, red, like that. Quantitative again multi-state with a number. Number of leaf 2, 3, 4, number of petal 5, 0, likewise. So, such characters should be converted into two state first. Then, how you will convert it? For example, flowers white or colored. What do you mean by that? Colored includes red, yellow, blue, likewise. So now you had converted them into two state. So those characters with a multi-state have to be converted into two state. Flowers white or colored. Leaves 2 versus not 2. Likewise you have to convert it. Clear? So the two state character you should code it as 0 and 1. So only two codes for a character. One is 0 and one is 1. Okay. Now this coded data is entered in the form of a matrix with t number of rows and n number of columns. Okay. So coded data is now entered in the form of a matrix with a small t number of rows and a small n number of columns. Columns include characters, other one includes number. See, this is the how it is arranged, characters and taxa. So these are the characters mentioned here. Here 10 characters are taken. So this is the taxa. Then code like is first character 1. In both cases 1. Both first character is present in the taxon A, taxon B. 1. If it is absent or opposite character, you can write as 0. Okay. Now, next step is what? Measurement of resemblance. So, now you have to calculate the resemblance between every pair of the operational taxonomic unit. The spelling is wrong, OTU. Every pair of OTU. Okay. A number of formula are there to calculate similarity or dissimilarity. So, different methods are, no need to go into detail. So, just mention simple matching coefficient, coefficient of association and Yule coefficient to measure the resemblance. So, one example here, simple matching coefficient. This is characters present or absent. Likewise, it is written number of matches 2. Number of matches 2. What are the number of matches here? This one and this one. See, in both the taxon it is present, this and this one. Number of mismatches, again 1, 2. So, what is the equation? M divided by M plus U. Okay. So, 2 divided by 2 plus 2, 1 by 2. That is the simple matching coefficient. Okay. Now last one is cluster analysis. How can you analyze in the form of a diagram? So, here we arrange the OTU in the order of decreasing similarity. So, find out resemblances, then based on that decreasing similarity you have to arrange it. Then you have to prepare what? Phenogram or cluster diagram. Two methods are there for preparing cluster diagram, agglomerative method and divisive method. Okay. So, what are the applications? Numerical taxonomy help in solving taxonomic problems, and Clifford performed it for 4 classes of monocotyl. That is the first one. Then Young and Watson used 83 attributes, characters in their analysis and 543 representative. Hilu and Wright undertook a numerical analysis of gravineous. Some very few works are related with. So, these are the applications of a numerical taxonomy in angiosperms. So, what is numerical taxonomy? From this portion, you have to just study the steps. Selection of OTU, then characters and its states, then resemblances. Based on the resemblances, prepare the phenograms. That's all. Then also study the Neo-Adansonian and Adansonian principles. That's important. Okay. So, that's all about the Adansonian method or mathematics or numerical taxonomy. Now, the second half is cladistics. That is phylogenetic method. Developing a classification based on phylogenetic data. That is phylogenetic analysis. Actually, the method is truly based on relationship, and diagram developing is called a cladogram. What we discussed in the last session, numerical taxonomy, that is phenogram. Here it is cladogram, cladistics. Okay. So, biologists who practice this method, you can call them as cladists. And this method was first developed by H. Henning, who is the author of phylogenetic systematics. So, here actually evolutionary relationships is documented. But, so very important one is relationship is measured here. And these methods, we construct a cladogram, and here another part also there, that is phylogram. That is a tree that contains evolutionary distance. So, ancestor is very important here. So, phylogenetic data are analyzed. That is one principle is applied here, that is known as parsimony. So, the maximum parsimony, like you can see in the modern molecular phylogenetics, these terms. What is parsimony? Likelihood. That is, most likely evolutionary route is the shortest pathway of changes that explains the pattern under observation. Evolutionary route, that is actually parsimony. So, the shortest hypothetical pathway of changes. Here actually we are imagining the ancestor. Then the shortest route we are selecting to reach the origin of the species. That is actually what parsimony. Then in the numerical taxonomy, our unit, working unit is what? Operational, what is that? Operational OTU here. Okay. Here what is that? Operational Evolutionary Unit, OEU. Okay. So here it include a hypothetical ancestor, its list. Then homologs are shared and derived characters were using here. There we used phenetic characters and morphological characters that we can see and we can observe and everything. So that's a mixing between empirical and observational taxonomy. But what here we are doing? Here we are imagining actually an ancestor. Then homologs shared and derived characters were using. So two types of characters are considering in cladistics. One is Symplesiomorphy, that is possession of primitive character. Synapomorphy, possession of derived character. So Symplesiomorphy means acquiring, possessing what? Primitive character. Synapomorphy means possession of derived character. Okay. So one example is given here. So see, what is this? This is the ancestor region. So from this, these points are called as node in the diagram. So here you can see that A, B, C, etc. are actually clades, different types of stalks. So character states unicellular sporophyte, multicellular sporophyte. So this is primitive one, first one. From this actually the characters derived. From the primitive one, the liverwort derived. Again from this conifers derived, angiosperm derived. So they acquired or derived some characters, new characters, that is a derived character. Okay, so here primitive and derived two characters are we considering. Okay. So how you can code it? There we studied numerical taxonomy, how we can code. Here how we can code it? List of characters prepared. We studied plesiomorphic and apomorphic characters, derived and primitive. Then determine the character state. Then find out the distance, measure of distance. A data matrix with coded character state for each evolutionary unit is used. So a data matrix is prepared and characters codes. Then cladogram is constructed. Okay, so here actually the character states of plesiomorphic and apomorphic, primitive and derived is considering based on that data matrix prepared and coded. And two methods are there for cladogram. One is rooted tree method and another one is unrooted tree. So rooted tree is actually we are placing a hypothetical ancestor taxon as basal member. At the base of the member there is an ancestral taxon from which all others evolved. That is the imagination. Okay, so that is a rooted tree method. See, this is a rooted tree method. What is the ancestor stock? The root. What is the root? This is the root. Root is the ancestral character. What is that? Ancestor with the chloroplast. Okay, now from the root, how many of them derived mosses? That is an outgroup, a separate one. Vascular tissue, a derived character fern. Seed, a derived character conifer. Flowers, a derived character lilies or modern. So, this is the final placed one. Then that is, you can call it as a clad. Okay, the state that is known as what? Clad. It reached a final position. So, these are the rooted diagram. Okay. Now another method is by Wagner ground plan divergence method proposed by Wagner based on apomorphic characters within the taxon. Another one is unrooted one, non-direction, no polarity character specified. They can be, that is no rooted, this is simply drawn. So we can convert them into rooted also. Okay, so stepwise transformation of characters. See, this is an unrooted one. You cannot find out a root here. Now, this is the Wagner tree. So that's all for cladistics. So actually what we studied in these two types is, one is numerical taxonomy or phenetic analysis and second one is what? Cladistic analysis. First one we prepared a phenogram, second one we prepared a cladogram. First one is strictly phenetic, morphology or observation, but second one is strictly what? Strictly the phylogenetic. Here we need an ancestor, an imagination. Okay. Then hypothetically we concluded it into, and in the first one actually what? Operational taxonomic unit. In second one we are evolutionary unit actually. So first cases also we are coding characters, usually 0 and 1. And the second case also we are coding, and in both cases we are finally make them into form of a diagrammatic chart. First one it is phenogram, second one it is cladogram. So in second case principle is actually what? Parsimony. Apomorphic and pleomorphic characters are considering, that is primitive and derived characters. Okay, so that is you can compare what is cladistic approach and phenetic approach, that both are very very important. Clear? So I hope you understood well. Thank you very much.